Thank you very much. Thank you very much for the introduction, Derek. And hey, everyone. Thank you. Good morning or good afternoon. Thanks for your time to attend the webinar that our company hosted this time. So the title of my presentation is Overcome the Hurdle to Generate High Affinity and Diversity Antibodies to Self or High Homology Antigens. So as we know that what will happen when we get sick. So this healthy person, and if we get infected by those pathogens, no matter the virus, bacteria, the pathogen will enter our body and to amplify, then we get sick. Thus, our immune system will be recruited to kill those pathogens or to recognize those foreign antigens. So two immune response, humoral immune response or cellular immune response will be recruited. The antibody is a major part of the humoral immune response. Antibody is produced by B cells. We all know that. Different B cells will make different types of antibodies and display the antibodies on the surface of the B cells. In our blood, only a very small number of those displayed antibodies will bind to the pathogen or foreign antigens that enter our body. Once the B cell recognize those foreign antigens, the B cells will be stimulated to expand and differentiation. The B cell that have been stimulated by the foreign antigen will differentiate into plasma cells and produce a lot of antigens. A single B cell will only secrete one type of antibody, and the multiple B cells each will secrete a different antibody, can code the molecule or the pathogen or foreign antigens. This is a theory basic for the monoclonal antibody technology. And what the antibody can do in our body? So the antibody firstly can recognize virus, bacteria, or even the cancer cell in our blood. The antibody tend to aggregate their targets, no matter it's proteins, virus, or cells. So once the antibody aggregate those targets, the antibody antigen aggregates may be ingested by phagocytic cells, for example, macrophage, NK cell, something like that, or the antigen and antibody positive cells or bacteria may be directly killed by the complement system with membrane attack complex holes. Sometimes antibody can also do bad things. For example, the GVHD from xenotransplantation or autoimmune disease, which the antibody can attack our own tissue. So what we can use for those antibodies, I think the most important application of antibodies is medicine. So a lot of therapeutic antibodies have been approved globally. And secondly, a lot of antibody can be used for biomedical research. A lot of benchmarks, such as Western blot, immunofluorescence, ELISA, whatever, we have to use antibodies. And we can also use antibodies for clinical diagnosis. For example, the COVID-19 test kit we use at home developed from those COVID-19 antibodies. And quite a few methods have been developed to make monoclonal antibodies. The conventional method is the hybridoma method. So the animal, normally mouse, will be immunized with the antigen, then the B cell will be isolated from the spleen of the immunized mice and fused with the melanoma, which normally we use SP20, and to get hybridomas. Then the hybridoma cell lines will be screened by the antigen, and we can identify the pathogen, the antigen neutralization monoclonal antibodies from the screening of the plates. Secondly, we can display the antibody on some surface of a cell or phage, whatever, and then we can screen the library with screen the libraries of antibody fragments in vitro. The animal can be human, can be mouse, rabbit, or even llama or fish. Sometimes people use shark. And this is a second method. Recently, people also developed some single B cell sorting using the fluorescence labeled antigen from the blood directly. Then RT-PCR or RNA-seq methods can be used to sequence the antibody sequence from those single cells. Those are three most used methods to clone antibody genes from the animals. Of course, the hybridoma method approach can provide high-affinity antibodies that have been matured in vivo, which is both cost and time effective. So for therapeutic antibodies, so we in the past 30 years, monoantibodies have been used for therapeutic purpose. At the beginning, people used mouse monoclonal antibodies. Then they found that it's very high antigenesis, it's very immunogenicity. So they just with the product was withdrawn very quickly after put on the market. Then people try to humanize, try to put more human fragments into the mouse antibody, which we have developed the chimeric antibody, human antibody, or even fully human antibodies. So the idea of the evolution of the antibody, therapeutic antibodies, is to make foreign antibodies better tolerated by the immune system of human, which to reduce the immunogenicities of those therapeutic antibodies. Currently, the majority of the approved antibodies are in those two classes, humanized or human antibodies. This is at least some examples of approved antibody drugs on the market right now. So as we know that, and right now, most antibodies are humanized, are fully human antibody. People at the beginning, people think fully human antibody should be the best one. However, if you compare the two PD-1 antibodies from Merck or BMS, so which one is better? I'm sure from business perspective, Merck's Keytruda is much better than BMS Optivo. Even BMS Optivo PD-1 antibody is a fully human antibody. I was part of the animal model group when I work at the BMS. So even now, the human anti-human antibody reactions are observed in most clinical trials. A lot of antibody blockbusters are humanized or even chimeric antibodies, even for now. For example, Keytruda or Herceptin, something like that. And also a lot of technology or even AI softwares have been developed for the humanization of mouse monoclonal antibody. So the development of a mouse monoclonal antibody is still a cost and time efficient way to develop the therapeutic antibodies. And in addition, the mouse monoclonal antibody is royalty-free. You don't need to pay a lot of upfront milestone, whatever costs to those companies who have the IP of the fully human antibody platforms. Like the mouse, I have been involved in two fully human monoclonal antibody mouse transgenic mouse platforms. You have to pay a lot of money to them. So that's a mouse humanization of a mouse monoclonal antibody is still a very good way to generate therapeutic antibodies. So for the no matter it's from the regular white type mice or from fully antibody humanized mouse platforms. So it is really hard to generate antibodies against self or highly homology antigens, since our body has a mechanism to eliminate those B cells that can produce antibody that can recognize self antigens. So all those cells during the B cell development, all those cells that can produce antibodies recognize self antigens will be induced to apoptosis. So unlike B cells that produce antibodies can recognize the foreign antigens will go through the development pathway and turn into mature B cells. So the target, since a lot of antigen, we want to develop antibodies against self or high homology antigens. So it's kind of a really headache, big hurdle for the researchers. Some methods have been used to overcome those hurdles. Firstly, the fusion expression of the self or high homology antigens with a high antigenicity T cell epitopes, so as to stimulate the immune system of the rodent. But for this method, you have to design the fusion protein very carefully, and the purification of the fusion protein is kind of complicated. And also, it's kind of complicated to characterize those antibodies after the immunization. The second way to get antibodies against self or high homology antigens is use some SLE-like autoimmune mouse models, for example, the NZBW strain. But this method has been universally successful since the antibody development from those mouse model, the affinity is low, and also it's a very high possibility that the antibody has some cross reaction with other antigens. So you have to be very carefully to characterize the cross reaction of the antibody with your target and also other antigens. Alternatively, mice with genetic knockout of a particular protein can be immunized with the protein so as to elicit the immune response. The lack of knockout animal for many targets of interest prohibits the routine application of this strategy. So I give you two examples how we can use knockout mice to develop antibodies against the self-antigen. This is the monoclonal antibody developed for mouse BCHE protein. So the antigen were the BCHE knockout mice were immunized with the antigen. In parallel, wild-type mice were also immunized with the same antigen. After 4 both injections, the antibody titer in the knockout mouse blood reached 1 to 50,000. And but for the wild-type mice, it's kind of a 0. No antibody indication, no antibody titer was observed in the wild-type mice. So this is for the bottom one, you can see that this is a Western blot analysis of the mouse BCHE antigen using Western blot. This is immunofluorescence staining of the BCHE positive mouse tissue. This antibody for mouse BCHE can also recognize rat or even human BCHE antigen, and no matter it's Western blot or immunostaining. So this is a very successful example to develop monoclonal antibody in the knockout mouse model. The knockout mice can also be used for DNA vaccine. This is the example of a prion protein antibody generation. So they knock out the prion protein gene from the mice and immunize the mice with 3 DNA constructs. The first one is a pre-protein PCG vector. As you know, PCG is a very commonly used vector for DNA vaccine. The second vector is a prior protein and P30, which is a TT antigen epitope. Is a very antigenetic and can be used to stimulate the T cell response, the immune response in the mouse. It's a fusion gene. And the third one is a blank vector, PCG blank vector. After 6 times of DNA immunization, you can observe very high titer of antibodies in the mouse blood in the knockout mice. Most of the antibody types in the mouse blood are IgG2A or IgG1, but no antibody was detected from the blank vector. In the white mice, even after 6 times of DNA immunization, you can fairly observe any antibody titers in the mouse blood. So the bottom picture shows the flow FACS analysis of the antibody in the knockout mice. You can see very significant peak shift with recombinant protein. And but for the wild-type mice, it's really the antibody titer is really, really low. This is the cell they used for the FACS analysis is N2A497 cells, which is positive for the mouse prior protein. So you can see that the knockout mice can be used for both protein or DNA immunization to get antigen-specific antibodies from those knockout mice. So as I said, as we know that there's a lack of all those targeted knockout mouse model, for our company, we have over 10,000 research-ready models. Of those 10,000 models, there are over 6,000 knockout mouse strains that are ready to use. So if you have your special need for any target, we can provide those knockout mouse strains for you to do the immunization. Then you to get your antigen-specific antibody development program in 6 months. So we can provide homologous mouse mostly in 6 months. However, even we have over 6,000 knockout mouse in our inventory, maybe you still have your own target, you want to look at it. So recently, our company have developed a new technology so to help you to generate homologous mice, knockout mice in 3 months. It's from gene ID from the very beginning to get 3 to 5 mice that you can start to immunize. So this is a new technology our company just developed recently. By this technology, you can do some immediate phenotype analysis or to immunize those mice with self or high homology antigen immunization. So the phenotype can be observed in those F0 mice directly. For basic researches, you can validate your hypothesis earlier and adjust your research direction swiftly. And also, this technology can enhance your research efficiency, which can reduce the number of the breeding generation and to expand the mouse population very quickly using IVF, and also reduce your cost, your time, and your resources. So how can we get to this technology? We can get homologous mice in 3 months. Firstly, we improve the nuclear entry efficiency, and we also optimize the transfection system, and we also refine our embryo manipulation technology. All those together, we can make homologous mice in 3 months. Here are 2 examples how we make those homologous mice in 3 months. This is a case study 1. We generate CR4 knockout mouse model in 6 months. So we knock out exon 2 from exon 2 to exon 7. And you can see here from the 7 new pups, we identified 4 homologous mice. Those are all F0 mice. And 2 heterozygous mice and 1 wild type. This is for conventional knockout mouse. For the case study 2, we generate CPA. We introduce a G2C mutation in the CPE1 gene. So we just this site where we introduce this mutation. This is a sequence analysis of this mouse after 3 months from the F0 mice. You can see here, this clear, this is a homozygous Paul mutation. So all those 2 lines, we got them in 3 months. Right now, they are all 8 weeks old. They are ready for immunization or for phenotype analysis. So for each kind of this project, we can promise to provide at least 3 F0 homologous mice per project. This should be good enough for you to do some antigen immunization, since the antigen you are going to use to inject is totally a foreign antigen for this mouse model, and which can stimulate very strong antibody immunoreaction in this mouse. So you don't really need to do 5 or 6 times immunization boost. So it should be short your immunization time. And you can do the genotype of those mice will be confirmed by typical molecular biology technologies, for example, PCR and sequence analysis. We provide health report and guarantee they are all free of special pathogens. We meet the requirement of ALAC, which are accepted by most vivariums in the United States. The universities, companies is okay with our mice. And we also provided you with a comprehensive report of this project. But of course, the technology has some limitation. For example, if the knockout of the gene can cause embryonic lethal or some early postnatal deaths, we cannot use this technology to generate homologous F0 mice. Also, if the gene can cause severe developmental defects and prevent the mice from surviving to the genotyping analysis stage, it is still unsuitable. So for those knockout mice, we can provide therapeutic antibodies for human disease in 6 months. This is a very timeline for the therapeutic antibody for human disease. We can provide the gene target gene knockout mice in 0 months. Right now, we have a lot of in stock, or 3 months use the new technology. So you can immunize the mice in 3 to 5 months. And you can either use a hybridoma, is a single cell sorting, or some surface display library construction method to characterize your leading antibody sequences. So the shortest time you can find your lead antibody sequences in 6 months. If you are developing therapeutic antibodies for animal disease, we can help you to get your antibody in 10 months. Why it's longer than human disease? For some of those therapeutic antibody for animal disease, for example, dog or cat, the majority of the antibody in dog and cat's blood are Ig lambda antibodies, not like that of a human or mice. For human and mice, the majority of our antibodies are Ig kappa antibodies. It's really hard to caninize or felinize those mouse Ig kappa antibody into Ig lambda antibodies. So right now, it's really hard for that. So if we use this new technology, we can help you to cross those target gene knockout mice with our Ig kappa knockout mouse to get double homo knockout mouse in 6 to 9 months. Then you can immunize those mice in 3 to 5 months and do either hybridoma cell fusion or single B cell and the surface display library. Since the kappa gene in this mouse has been knocked out, you can only get Ig lambda antibodies in this mouse. So it'd be easier for you to do the canonization or felinization to make antibodies for those pets. So for Genobiotx, our company is a one-stop animal models. We provide one-stop animal model services. No matter it's custom model services, including knockout, conditional knockout, knocking, permutation, target overexpression, or random transgene. We also, as I said, we have over 10,000 off-the-shelf mouse models you can choose from. And we also provide pharmacology, pharmacodynamic services, and other supporting services like the monoclonal antibody development services. And we are well-known contract research organization, CRO company, to provide those animal model services. And here's the history of the company. It's a first company established in China, specialized in the development of genetic engineered mouse models in September 2000. And our company went public 2021. Also, our US company called Genobiotx was registered in Houston, Texas in 2021. And we also have our office. We recently established our office in Boston area, which is located in the Cambridge Innovation Center. And these are some of the facilities of our company in Shanghai, Beijing, Zhongshan, and this is Houston and Boston. We also have a business development office in Europe, which is located in Oslo, Norway, and Japan is Osaka. I think that's all for my presentation today. Thank you very much for your time and attention. We are open to, if you have any question, we are glad to answer your questions. Thank you.